Hello, everybody. I'd like to welcome you to the BIH Lecture series. I have the great pleasure and honor to welcome Katy Börner to our series. Katy started her career in Leipzig and has a Master of Science in Electrical Engineering. She went on to get a PhD in Computer Science in Kaiserslautern, and wound up at University of Indiana in the United States. She holds a named chair, Victor Inge Distinguished Professor of Engineering and Information Science at the Luddy School of Informatics. She's the founding director of the Cyberinfrastructure for the Network Science Center. All of this at the Indiana University in Bloomington in the US. And she's done quite a number of things. I'm only going to highlight two of them. So she's the curator of the Mapping Science exhibit, um, and she leads the International Human Reference Atlas effort. I also have the pleasure of starting to collaborate with Katy over the last year, and I think we might hear a little bit about that. So, Katy, welcome very much to our series. I'd like to give you the floor.
Peter, thanks so very much. Nice to be here at 6 a.m. Bloomington, Indiana time. I added a virtual background here because it's pretty dark still out there, but very nice to be here. And I will share my slides, and if I do it correctly, I can also show you some videos. So here we go. Can you see my first slide? I assume so. So again, it's a great pleasure to present this work here. It's really collaborative work with Peter Robinson's team. Ibukia Apte is also here in the audience. She is a key player and has been a driving force for this new bridge between our team creating the Human Reference Atlas and Peter's team leading the human phenotype ontology. Let me tell you a little bit more about the Human Reference Atlas. Some of you might be familiar with this effort. You see not only me here, but more than 300 experts that have contributed and are now using the Atlas. These experts come from all kinds of different consortia around the globe. It's interesting to note that some of these efforts focus squarely on brain. That's a complicated enough organ to be busy with. Others focus on lung, and I will have some lung examples later on. Some focus on kidney, and again, on the right-hand side here, you see how many efforts there really are and how many organs they care about. And of course, the number of vital organs is long, and then there are many, many other organs which are also very much needed to be healthy and happy. And all of this is supported extensively by the National Institutes of Health, but also by the Human Cell Atlas effort and by many private foundations also, which we love to see. On the next slide, you will see that I want to give you a brief introduction into the Human Reference Atlas for those which have not heard about it yet. The Atlas will have to cover the human body across temporal and spatial scales, from the whole human body, it's a meter scale, down to the nanometer scale for the biomolecular elements that make up our bodies. And we capture this information aligned to existing ontologies using so-called ASCT+B tables. AS are anatomical structures, and so you see a parthonomy for the kidney on the left here. Then there are cell types, and there's a typology in cell ontology that helps organize these cell types in more and more general groupings. And on the right-hand side, you have biomarkers. And for the Human Reference Atlas, we capture genes and proteins and lipids, metabolites, proteoforms, and other biomarkers. And they are ultimately also in a typology tree. And these three trees, they are interlinked via located in and characterize linkages. So there are biomarkers which are used to characterize cell types, and these cell types exist in certain anatomical structures. So it's a quite complex structure, and data visualizations really help here experts to not just construct, but also review and ultimately use these ASCT+B tables. We spent the last 7 years to agree on terminology. I think it's still not totally the case that everybody is using Atlas or Common Coordinate Framework for the very same concept. It took us quite a while to agree within the Human Reference Atlas on those terms and many, many other terms. So we hope this glossary is also helpful for other atlasing efforts and ultimately also for teaching the next generation of clinicians and doctors and others. We are constructing the Human Reference Atlas by using human experts, which by hand, based on what they learned and what they have seen on the operating room, create these ASCT+B tables. We can have medical illustrators create these 3D references and also 2D references for functional tissue units. We work with machine learning experts to devise new spatial segmentation, training data, and models. We have another entire working group on organ mapping antibody panels, agreeing on antibodies that can be used to understand healthy human tissue across assay types, tissue preservation methods, and also across organs, of course. And each organ is quite different from each other. And of course, oftentimes there are different ambitions that the organ mapping teams have. And then there is the entire area of cell type annotation tools for single cell transcriptomics data. And there are many, many tools available now. For the lung, you have 7 different cell type annotation tools that are widely used, and probably many more that are also used. So it's not always known which one is best for which organ. Down here, you see that on our end, we then use all that data and compile HRA-POP data, cell type populations per anatomical structure. So here for the heart, for instance, you have different types of cells and different numbers of cells of those types in the different anatomical structures. And we also use large language models to analyze literature to get for instance, the size of functional tissue units extracted using large vision models applied to PubMed publications, for instance, or to also understand which cell types are commonly located and associated with which anatomical structures to ultimately validate the ASCT+B tables. But we also have looked at who is funding which organ, which experts exist for which organs, so that we can send out the digital objects that make up the Human Reference Atlas to the right experts around the globe to ultimately arrive at a community consensus at an international scale, not just focused in on one country or 2 countries, but really globally. Here you see some of the 3D reference organs. You only see the outer shell of these organs. Many of them have substantial number of anatomical structures inside. Here, for instance, you have the kidney with many of the very important internal structures. You also see down here below a very exciting collaboration with dentists. We modeled more than 30 anatomical structures for them, and they are very interested to also bring in data on the human microbiome because that's very relevant for oral health and ultimately the health for the intestine and many other organs. Here you see a screenfall of functional tissue units. Typically, you don't get to see them all at once. Here you get to recognize that some of them are at quite different spatial scales. And we now also have the hair follicle and some other very large anatomical structures that are all needed to be healthy. And all of this data is available freely, can be used in textbooks and in teaching, and is now also available via the NIH 3D site and the NIH BioArt website. Ultimately, you can access all of this data programmatically, but also via this KG or Knowledge Graph Explorer. So you can, for instance, search for your favorite organ, and you get to see all the many digital objects that now exist. And again, all of this data is licensed in a way that you can take it and make a company from it. So we want these kind of derivative artifacts and applications that benefit from this community effort. I wanted to show you a video that hopefully drives home the enormous scale we are having to deal with at the spatial scale. This is not yet capturing the temporal scale, which also goes from below second to lifetime, which is also another major scale difference. But here, let's zoom in from the meter scale down to nanometer and back out. You might be familiar with the Powers of 10 movie by the Ames partnership. Here you have a similar zoom, but using the Human Reference Atlas data, which again is now freely available for anyone to use. So let me play this, and I hope you get to hear the sound. So you might have seen some of the images. Those are extracted from PubMed papers and arranged in mosaic or in the spheric layout. You saw the network of single cells. This is actually GTEx data. So for everything you just saw, there's code and data that is freely available. And in fact, we have been making this available to artists also so that they can show what they would do with this data. And of course, it's also available to all of you if you are interested to access this data. So if you go to this website, then you will see full provenance for everything that was just in the video. So another use of the HRA is, of course, to start predicting what types of cell type populations might exist in certain areas of the human body. So you might have a colon, for instance, and you take a biopsy data set, and you would like to know what is healthy normal in a certain part of the colon. And we know that cell type populations actually change as you go through the colon from begin to the very end. And the Human Reference Atlas, thanks to the Human Reference Atlas populations data, you can start doing these predictions for certain demographic groups for which sufficient data exists. You can also take a data sample for which the location in the human body is not known, it was not captured, and you can start to predict where in the human body this tissue block might have been cut from. And so the way this works is that you would, for instance, if you just cut a biopsy data set, you would register it into the registration user interface. Then you might have this 3 tissue blocks that gets either run through light sheet. In this case, you don't have to cut it into 2D tissue sections, but oftentimes in order to run immunohistochemistry and other assays, you would cut it into these 2D tissue sections, which you might later on 3D reconstruct again and segment and annotate it. But then you might also run transcriptomics analysis, single-cell RNA-seq data sets, where you have more information on the single cells from all the genes that are in there. But you also might run proteomic imaging or H&E stains. And oftentimes, ideally, you have H&E for all of your spatial data sets, because we can use the H&E stain data to segment out the functional tissue units and then analyze the cell type populations within those functional tissue units. And we are focusing in on these FTUs, as we call them, because they are delivering main functions. They are the alveoli in the lungs that get you oxygen, or the glomeruli in the kidneys that do the filtration, or many, many other functions that exist across these vital organs. And so all this code, which is available to do the reconstruction, but also to run the atlas, is available via this recent Nature Methods paper. And it's a total of 50 algorithms, which all have to work together. So this is a huge community effort, again, to agree on API interfaces to make this work. But after this is done, you can then take transcriptomics data and run it through 7+ different cell type annotation tools, get the azimuths or other reference maps, agree on annotated cells. And as you know, some of these cell type annotation tools assign quite different cell type annotations to the very same cell in the same data set. So that's up to algorithm comparisons and up to preferences also partially, which cell type annotation tool to use. In HubMap, we use Azimuth from Rahul Zahid's team. It's a New York Genome Center. If Azimuth is not available for an organ, we use Cell Typist by Sarah Teichmann. And if none of these two can be applied to an organ, we use PopV. So that is a massive effort by the John Zuckerberg effort to create an Homo sapiens atlas ultimately across many, many different organs. If you have spatial data, for instance, immunohistochemistry data, then you might be interested in the 27 OMAPs. These are organ and tissue preservation method and assay type specific listings of antibody panels together with suppliers for those that can be used to annotate cells in healthy human tissue. And these OMAPs, again, are freely available. It's a huge community effort. And based on these OMAP-based panels, we can then also map the data to the Human Reference Atlas. After this is done, you can then again take a tissue block and start predicting what cell by biomarker expression values you will most likely have in these areas. And the biomarker expression values could be gene expression values or protein expression values. Or you take an existing cell by gene or cell by protein matrix and you start to predict where in the human body this tissue might have come from. And oftentimes you get back corridors because there might be two anatomical structure on which this tissue block writes, and then it's just a longer area. Here in the middle, you see a comparison of healthy lung to diseased BPD lung. And you get to see that these distances from cells to vasculature actually change when disease comes in. And in fact, you can also use this to characterize disease progression. And I will have some more slides on this later. Down below, you have a very exciting work by John Hickey's team, formerly at Stanford University, now at Duke. And he is looking at hierarchically nested functional tissue units. And you see here an entire hierarchy of very detailed functional tissue units, and then larger, more generalized neighborhoods, and then cell types, and then even more general cell types. And we believe that these cell neighborhoods are actually the ones that generate the function and that also change as disease comes in or when senescence comes in. For instance, there's an entire effort on senescent networks, SENNet, which looks squarely at where we can find and how we can characterize senescent cells. So if you are a lung expert, you might like to zoom into lung. So you have the body, from the body it goes to the lung itself, and then other systems that are relevant for lung. Here you have the autonomy for the lung. Then you have the many cell types that you can find in the lung, and then you have the gene circular and protein, these diamonds, diamond-shaped elements that are used to characterize these cell types. And all of this is available online. You see that there are a total of 54 anatomical structures with 74 cell types and 333 total biomarkers just for the lung. And there are currently 34 different organs, unique organs in the HRA. And if you count male and female separately, then it's 72 organs. So here you have the male heart, kidney, and lung with the tissue sections that have been registered into these organs. And again, all of this can be explored via the exploration user interface. And you can, of course, make more organs visible or make just your own organ visible. And you get to see which other teams have been registering tissue in support also of meta studies. And ultimately, no organ can perform their function alone. So you actually oftentimes need to look at multiple organs to really understand function of the human body. We also have an HRA organ gallery, which is a 3D virtual reality environment, which is freely available on the Meta store. So if you happen to have a VR device, please feel free to download it and explore this data in 3D. And here you also get to see this enormous zoom from the whole human body down to the protein structure level. The Human Reference Atlas cell type population effort has a bioarchive preprint out now. We also just yesterday submitted the revised paper. So fingers crossed it just gets through this time. Here you get to see the different anatomical structures, and we always plot the organ name, the number of data sets, and then the anatomical structure. And here on the left for male and on the right for female, you get to see the cell type populations aggregated at the highest cell type. Obviously, we have a hierarchy of cell types, and there are many more very detailed cell types that also exist for each one of these anatomical structures. And again, you can download that data from the HRA-KT Explorer. If we have data that allows us to zoom into functional tissue units, then we can extract cell by gene matrices for just the functional tissue units. Here is the bronchus submucosal gland. And if you have data sets from healthy versus diseased, you can start comparing what changes in terms of biomarker expression values, but also in terms of cell types as disease comes in. If you have data for different age groups, which oftentimes is available, then you can select young and then older and older and older to see what changes again as we age, and this is healthy aging. And again, you can browse through these 27 different functional tissue units across 12 organs to start to understand what changes within these functional tissue units. Coming over to the cell type distances, this is a tool which is now widely used by different teams. If your data can be rendered in a format so that you have X, Y for 2 spatial dimensions, and if you have 3D data, you can also add a third dimension and a name, either a cluster name or a cell type. Then you can read that into the Cell Distance Explorer tool. And you will get to see beautiful renderings of your data. You get to really see your data in new ways. Here's a zoom into the intestine. And you will also get to see cell distance plots. Most of these cells have to live in the vicinity of 200 micrometers to vasculature to get the oxygen and the supplies they need. But of course, if you have a 2D section, then some of the connections might be to the next layer up or down. So it's always better to run this analysis using 3D data, but 3D data is still very expensive to get. We have applied that now to 14 studies with 12 different tissue types, and you see all 12 of them shown here. This is a study that runs over 47 million cells, and that is also available on bioRxiv together with links to all of this data. And also much work has been gone into harmonizing these cell types across tissues, across assay types. And so again, this harmonized data and the cell type typology is also available via this paper. Ultimately, you get to also use the Cell Distance Explorer on other existing data that has been published previously. And so here you just get a few visualizations of new views of the human body, I would call them. And in some cases, there is colorectal cancer here, so disease in the data. It's not just healthy data that you can run this on. All right, in the interest of time, let me continue with the vasculature common coordinate framework. So ultimately, we believe a Cartesian coordinate system will not suffice to map all of us onto one reference framework. So there is an extra effort going on to look at in how far we can use the vasculature, which is already multiscale, from the rather larger aorta coming out of the heart down to the microvasculature that goes everywhere to our fingertips and everywhere else, as a way to have a latitude-longitude system for the human body. And I won't be able to show this video, but this is also available on YouTube. So if you look at vasculature common coordinate framework on YouTube, you will get to see a video that compares how a package is delivered using the postal latitude-longitude system. In the US, it's the zip code. And in comparison with the human vasculature, that has ultimately not just biomarker expression values, but also morphological features, branching angles, and diameter of vessels to help us understand where we are in the healthy human body. So let me go over to the very exciting new work, which is Human Reference Atlas and Human Phenotype Ontology combined. And again, this is work with Oliver He's team, who is at the University of Michigan, who is working on kidney. And then, of course, the Human Phenotype Ontology team, which is led by Peter Robinson. But ultimately, the superpower here is really Aybouki Alte, also here on the call. And it's really neat to see this all come together. And this is with support by Stiftung Charité. It really creates a network across our 3 teams. And we had a lot of fun comparing the holdings in the Human Phenotype Ontology here on the left. And I put the link here in case you had not have a chance to check it out. And the Human Reference Atlas here on the right-hand side. And we are interested in creating a bridge, but then also showing that it makes a difference on kidney, and that's where Oliver Heath's team comes in. So for the Human Phenotype Ontology, you might be familiar that it represents the rich diversity of human phenotypes and ultimately enables computational inference over these combined genomic and phenotypic data sets. And it's widely used in clinical praxis, and it has over 19,000 terms to represent also clinical manifestations of disease. It's embedded in a larger knowledge graph, which is wonderful for our purposes because the HRA is also encapsulated in a knowledge graph. So by creating a linkage between the HPO knowledge graph and the HRA knowledge graph, we can then run a lot of new analysis, which were not possible before. And the Human Phenotype Ontology is useful in support of genomic diagnosis, gene disease discovery, genotype, phenotype analysis, mechanism discovery, and many, many other analysis which are relevant for clinical praxis, but also for research. And so again, the linkage to HRA then provides spatiality to where certain genes are when disease comes in, but also provides new user interfaces for understanding and using the human phenotype ontology. So it's a win-win. So we had a number of hackathons. The first one brought the team from the University of Michigan, here Oliver He and Jing Zhang, but also Aibukya Atta to Indiana University. So this is a photo taken in Luddy Hall. And of course, we had good food. We went out on a sailing trip. It was wonderful to have everybody in person here. But we also got a lot of research done. And ultimately, there are now first interlinkages between the Human Reference Atlas knowledge graph and the human phenotype ontology, but also to the KPMP kidney disease atlas knowledge graph. So there's a third knowledge graphs that can now be mapped in as well. And so if you interlink those three knowledge graphs, then a lot of new insights can arrive. And we are in the process of starting to analyze that data, that combined data, and to see in how far this really creates new insights to how disease unfolds, but also in how the spatiality, which we cover in the Human Reference Atlas, can be used to augment the analysis that has already been done with human phenotype ontology. And of course, the knowledge graphs that you just saw, which are out of these three knowledge graphs, can always be expanded by adding links to other bioinformatics resources. For instance, HubMap and ZenNet and also the Human Reference Atlas are part of the common data ecosystem. And that effort, which combines the data from 14 NIH-funded Common Fund studies, is also captured in the CFDE knowledge graph. So that links over to a lot more data that might be relevant for understanding health and disease. And of course, all of this is then ready for AI, machine learning approaches that can then be run over that data, can be trained on that data, and can start to predict data with provenance back to the rich data that exists in the existing knowledge graphs. So for instance, Robinson's grape library for graph representation learning can be used to predict links between anatomical and cellular entities and disease manifestations. And so you have the references to these relevant papers here on the lower right. What we have been doing concretely also is to look at the 3D anatomical structures here in male and female. You see that they're slightly different. There's even a slightly different number of renal pyramids. And you can now map the gene expression values back into this 3D context, and you can start to see what happens as a disease comes in within these 3D structures. You can also use the cell distance explorer we just looked at to understand what cell types are most affected by specific clinical manifestations. So here we are using the rich HPO terms. We are also interested to see if cell types related to specific HPO terms differ between associated diseases, so different types of cancer, for instance, different stages of cancer. We are also interested to see if the distribution of cell types changes as clinical manifestations of diseases develop, and also see if in how far we can identify disease stages, and ultimately to stratify patients so that they get the best possible treatment, given that oftentimes they need the right treatment immediately and not a trial and error. Ultimately, we are also very, very interested to see how the distribution of gene expression and splicing changes in diseases, but maybe also in aging. And we are curious if these patterns can be used prognostically. So ideally, we would catch diseases earlier, we would stay healthy, we would have precision health in addition to precision medicine. And so again, here you have a data set by Peter Sorger's team loaded into the Cell Distance Explorer. You have tumor cells in red, so you get to see where the tumor is manifested. And then you have the endothelial cells in this light blue, and you have the distance distributions to the cell types, and you see that tumors absolutely connect to the endothelial cells. I would like to thank the entire team. This is the HubMap team that is part of the MCIU team, which is part of the Hive data core and engagement team within HubMap. And then HubMap currently has 350 researchers, which some of them made it to a beautiful all-hands meeting in California. So you see us under palm trees, which were cropped off. But then there's also CENNET, which is another 400 experts. There's KPMP, probably another 100 experts. And again, all of these consortia, these are big science projects. So this really cannot be done by one person or even a smaller team. It's a huge community effort. And we hope that the community benefits from all of it. There's also a lot of fundings that we did benefit from. And I would like to thank all of the funders here. There is another slide which links again to these two resources, the Human Reference Atlas and the Human Phenotype Ontology. Here are the emails for myself and Peter, and we would welcome questions, and that's the end of my talk. I'm happy to run the video if there is more time on the vasculature common coordinate framework, but there might be questions. So let's see first if there are questions.
Yeah, thank you very much, Katie, for a wonderful talk and incredibly impressive work. We do have a first question, but I'd like to encourage the rest of the audience to enter any questions you may have while we're talking about the first one. So the first question was sent by Oliver Klein. And so he's saying that similar cells can exhibit different behaviors or properties depending on their environment. And he'd like to know to what extent cellular interactions and properties depend on the environment and how that's taken into account in the atlas. So not an easy question, but
yeah. This is a great question. So HubMap has been focusing on spatially explicit data. So that's why we also focused on these OMAPs so that different teams can benefit from each other's antibody panel designs, because each antibody panel design is like a postdoc for a year and lots of trials and errors to get a good panel. As you know, there are other data portals like the Shansaku-Berg cell by gene portal, which focus on transcriptomics data. But if we go to spatial data, then of course each cell that's segmented and named inside of a tissue section, and some of these tissue sections are large. You might have up to 1 million cells, 500,000 cells in one tissue section. So these are really large data sets. And then if you have a stack of those, then you have multiple of those in a 3D reconstructed data set. And so within this 3D reconstructed data set, you would have a cell that lives in a context. And the context absolutely matters. If you are near a senescent cell, it's not a good neighborhood for you typically. So you want to be part of a productive team, of a healthy team, of a functioning team, and not part of a team that is deprived of oxygen or has other problems in life. So what we have been trying to do is to identify teams of cells that work productively together. And one way of doing this is to look at functional tissue units. So inside of each functional tissue unit, you would have certain numbers and types of cells, and they all have a very well-defined spatial composition. And in fact, if the capillaries are not connected in the right way, blood will leak, right? So it's actually very well defined. And it's the same for the glomeruli, where urine has to also go through. So it's a very well defined spatial structure. And depending on your environment, you will do different things. Absolutely. And there are beautiful demonstrations by Michael Levine's team at Tufts University of what happens if you put cells in a new spatial context. they will do something quite different. They are agential materials. But coming back to HubMap, if you now have that cell in a 3D spatial context, you can then start to see what happens as we age, what happens as disease comes in. And again, these cell type gene expressions, but also protein expression values will change as you are encountering a different environment. We have also been watching closely all that really amazing work on cell neighborhoods, on cell niches, etc. Oftentimes, people start doing random sampling across spatially explicit data, oftentimes 2D tissue sections. But what you get there is that if you have a functional tissue unit, let's say the inner part of this circle here, and you look at a sampling circle, which is my left hand here, that's overlapping the functional tissue unit and the outer context of this functional tissue unit, then most likely some of the cells that are inside, they are very much working together as a team. But the ones which are outside here, which are not part of the functional tissue unit, they might have a very different function. They might hold everything in place, which is important. They might supply blood and oxygen and other parts to the FTU. But they might not be able to really be part of that teamwork internal to here. So I think by looking at which functional tissue units, ideally nested, you saw John Hickey's approach there, are really delivering specific function, that might be our best way for truly understanding not just the structure of the human body, but also the function.
Thank you. Well, I have a question I'd like to ask. So one of the things that, our group has also been working on a data resource, a knowledge resource, but looking to what you've done, you've made this incredible community and gotten so many people involved in this project. I was wondering if you could just tell us a little bit about the history of the project and what you think the recipe for your success was in just getting so many people to come together.
Great question. So it's absolutely a socio-technical engineering feast. So it's not just building a technical infrastructure. It's really also listening of what people need and want and can do and what's feasible with the existing technologies, assay types. As you know, single cell studies are still very, very expensive. About 8 to 10 years ago, there were two major lines of research and ambitions that came together. One is the Human Cell Atlas effort led by Aviv Rajev and Sarah Teichmann. And they have been leading that effort masterfully, really, since all these years. And I hope you get to go to the Human Cell Atlas community meetings where thousands of experts really collaborate together around the globe, also bringing in rare diseases and other data sets to basically focus on the human cells and create a cell atlas. And then the other line of research and ambition is Zorina Gales and many of the NIH experts realizing that there is an opportunity to fund HubMap and similar efforts to map the human body at scale as a common fund project, and to then put out calls for proposals and to get tissue mapping centers funded. And these tissue mapping centers, they were funded earlier than the Hive team, which I'm part of, because it takes time to set up these infrastructures, to hire the postdocs, to buy the equipment, to get first data to come out. And then when the Hive team came together, and there are 6 teams, there are 2 mapping centers, Rahul's team, it's the New York Genomics Center, my team, 2 tool development teams, and then also the ISE at Pittsburgh and CMU, bringing together the supercomputing center resources also. And then a new team that was recently funded at the University of Florida. And so this HubMap team really has financed and promoted the initial data sets and structure and working groups that were needed to envision something like the Human Reference Atlas, but then powered by the Human Cell Atlas, it became possible to really reach so many experts and to engage them and to have them not only construct these ASCT+B tables and OMAPs and AVLs and many other human-generated artifacts, but also to serve as expert reviewers. And again, you ultimately have the experts creating it, then you have experts reviewing it, then you have large language models also validating it, and then you have the experimental data, which can also be used to ultimately validate the data in the Human Reference Atlas. So it's a huge community effort, and I have made friends which I never knew I would make. And it's really enriching my life also. So it's a really wonderful effort.
Yeah, no, it is truly amazing. We have a question from Abdul Ifat. So thank you. Abdul is an AI researcher and a physician from Hamburg, and he wanted to ask if the models trained with the data you generate are available as well. And he's asking how those models would be trained and what we think the outlook of those models on predictive analytics might be.
Great question. Thank you. So we run, as part of HubMap, 4 Kaggle competitions. Actually, one of them was funded by CENNET. So 3 funded by HubMap. Each one is a huge effort because you have to create a data set that the community can use. And we focused on FTU segmentation and vasculature segmentation there. And then CENNET also funded one. And these Kaggle competitions, they were in close collaboration with experimental teams because we needed data that has not been published for evaluating solutions and that can be held unpublished for a year, which is also hard for many experimental teams. One of the Kaggle competitions was also in collaboration with the Human Protein Atlas. Emma's team, and Emma Landberg is now at Stanford University also. So there are great collaborations across the globe. But in these Kaggle competitions, you have, of course, all the data available online. All the models are available online, and also the metrics. So you could start taking your own model and start comparing it with the other models that were the winning models that actually won the prize money. But you can also then run it through the same metrics that we created together with the Google team for these 4 Kaggle competitions. For the cell segmentation and annotation, we work closely with the team by David Van Welen at Caltech, and he is making his data available. His CellSum paper was just accepted, so it should come out any moment now. And so that also always comes with the trained models and the data that was used to train. So again, we would like this to be as open as possible for the community. I know there are many other teams which have to hold back data because maybe it cannot be shared, which have to not publish certain trained models, but that's not what we decided to do.
Thank you. One other question, I guess, about education. So I wish that HRA had been available when I was studying anatomy and histology in medical school. What are your plans to sort of deploy HRA as a part of medical education, or are people using it already?
Great question again. So we have been creating a so-called visible human MOOC. So if you go online and just search for that, you will see a massively open online course which details how the data comes into existence with kind of walkthroughs, experimental labs, so that you really get to see what does it take to do OMAP panels, right? How much work is this and how do you go about it? Or how does Rahul Zahid's team create azimuths, which then can annotate so many cell types across so many tissues? So Rahul himself explains that to you. We also have our medical illustrators explain how they created the 3D anatomical structure models. And so we hope this is valuable for you to understand how the atlas is constructed. But then going forward, we are also actively looking for collaborations to bring these new data objects into classroom settings, but also into textbooks, also into videos like the one you saw that potentially can help the next generation of researchers, but also clinicians understand that enormous scale difference. Oftentimes, surgeons are trained on the anatomical scale, pathologists are trained on the H&E scale, but the functional tissue unit scale, there's actually not so much information. And in fact, we are still trying to get our hands on 3D data to segment out the functional tissue units in 3D. So if you have that kind of data, please reach back out to me. Because it's a very important stepping stone between the large size of human organs, 10 centimeter kidney, very, very long intestine, and the single cell level, 10 microns. And we need to bridge that scale because ultimately the function happens at these teams of cells level. And so ultimately there are some first collaborations emerging where people have guest lectures, where, for instance, Andreas, who leads the Human Reference Atlas organ gallery, comes into your lecture and just shows you what data now exists for these different levels and what kind of insight you can take away from it. But we would like to have it used even more in the educational setting.
Super. If people want to find out more information about this, would they go to the HRA website, or where should they go to
Yeah, if you go to humanatlas.io under training, you will get to see the Visible Human MOOC, but you will also get to see, I don't know, 60 SOPs, which are standard operating procedures of how the atlas was created and can be used. And you will get to see scrolly stories, which I should also mention here. So these are websites where you just go down the website and you get to encounter different animations and stories, which explain to you, for instance, how a cell can learn to find his or her own name using the ASCT+B reporter. It's actually very cute. So ultimately, this is meant for very general audiences, and it's supposed to be also fun. I think we want the exploration and this fantastic voyage of the human body to be fun and to be exciting and invigorating and inspiring for the next generation.
So we just have a few more minutes. Maybe I'll close with one final question. If anybody else has a question, now would be your last chance. Were there organs that were particularly hard or easy, and why?
So when we started to compute these cell type populations, it became very quickly clear that there are some organs where tissue probes are always taken in the same area. So there's a systematic process going on to, for instance, take ultrasound-guided biopsies from the kidney cortex in a very systematic way. And then as time progresses, you get to see always same or similar cell type populations, definitely for the same demographic group. So we have a plateauing of the data. So maybe don't sample there or start sampling in other places. Or well, ultimately, there is now a very good signal for that area of the human body. However, there are also other organs where every single time you take a sample, it looks like there is a quite different cell type population in terms of how many cells of one cell type exist, but also the gene expression markers. And it could be that the assay types are just too different, or the donor demographics from which it is sampled is also too different to really make it comparable across the data sets. But it could also be that these organs have more day-night cycles or hormonal cycles or other elements going on that we don't yet understand. And so when you look at the HRA-POP data sets, you will see that some are converging and it becomes very robust as a signal. Whereas others, I think we need more data and maybe also have to sit back and try to capture other elements, like for instance, for the lung, we now ask that we also get information on smoking or not. BMI, if you can capture it, seems to make quite a bit of a difference. So please do add it to your donor demographic metadata. Menstrual cycles, post-menopause, all seems to make a difference. So please try to capture that data, because it will make a difference in how we stratify these donor populations for understanding healthy beings.
Well, thank you very much, Katie. I think the entire BIH community appreciates that you got up pretty early today. Wonderful lecture. We're all looking forward to more interactions with you over coming years. I would also like to take the opportunity to announce the next BIH lecture, which will be on January 30th by Andrea Ganna, who is an associate professor at the Institute for Molecular Medicine Finland in Helsinki, and an associate at Harvard Medical School. So yeah, thank you very much, and have a great rest of your day, everybody.